Good day, everyone. Welcome to our channel on the brain. Today we will discuss cognitive networks and how exactly can we model these networks. This video is like a prelude, if you will, to the coming videos where we will discuss the impact of digital technology on the brain, as well as the impact of large language model use on the development of the brain. We will present data in these two videos, subsequent videos, based on studies that use connectivity as techniques to characterize these large cognitive networks. And I thought it was a good idea to start with a sort of an introductory video that will depict the different technology we use to characterize these networks. So let's start without further ado. And let's start about a Canadian psychologist called Donald Houde, which has actually put together what we call the Hebbian theory, which basically states that, and I will cite him directly, those cells that fire together wire together. So what Donald Houde meant by this catchphrase is quite simple, actually. Means that if neurons discharge together and emit signals that are synchronous, they are likely to be connected and part of a singular neural network. To this day, we still use this concept to try to characterize cognitive networks. We use a mathematical theory that is quite old, actually, which we call the graph theory. Now, graph theory simply uses two different objects, so nodes as well as edges or links. So you have different nodes connected to each other, and using these two objects, you can characterize something that is quite complex in terms of networking in a simplified manner. So let me give you an example. This is a graph depicting the flight paths for the company, the airline Air Canada, in departing from Canada. So you do see nodes, large nodes such as Toronto, which is much larger than Moncton, as an example, that simply states that this node is more vital to our network. There's more departure and arrival in Toronto than there are in Moncton. So using this type of process, we try to model the large-scale cognitive network in the brain. Now, to do that, we infer that cognitive processing are produced by groups of neurons, which obviously makes sense, but interestingly has never been convincingly entirely demonstrated. So it is a presumption that I think is reasonable, but nonetheless a presumption. So we stipulate that there are groups of neurons that are connected together and that will, in their collective behavior, produce cognitive functioning. So now to highlight these groups of neurons and their connections, we're going to use two different techniques of MRI. Now, MRI has changed, has evolved, and is still evolving. MRI is a way to usually study the brain anatomy, but there are other things we can study by MRI, and it's all to the merit of those scientists that actually think about these fancy ideas. One such example is what we call diffusion MRI. So what does measure diffusion MRI? Your brain is bathed in liquid, as you know. You do have a lot of fibers in your brain. There's water molecules surrounding these fibers. So diffusion can measure the movement of water molecules. We stipulate that water molecule movements is constrained by the structure of the brain. Hence, if the molecules move in a certain way, we can presume that there's fibers, there's a material limiting the movement of water. So using this movement that can be measured across voxels gathered in the brain, mathematicians will use algorithms to translate these movements and connect the dots, if you will, to try to reproduce a model of these brain fibers. And this is what we call tractography. So using tractography, we can simulate the connections between neurons. So that's technique number one. Technique number two is called functional MRI. So functional MRI use another physical principle that has to do with blood vessels. Your brain is always active, but this activity in your brain is extremely heterogeneous and is entirely dependent of what task you're performing. Right now, I'm speaking to you, and my speech centers are highlighted. They need more blood, they need more energy. So the blood vessels in the area serving these specific centers are dilated, and the oxygen is more extracted in this area than in the areas for physical exertion. Let's say I'm sitting on a chair right now, so these are not too actively stimulated right now. So the extraction of oxygen and the dilatation of blood vessels can be captured by the MRI, the functional MRI, and this can highlight specific neuronal centers activated during a task. This is what you see on this MRI, where statistically speaking, these areas are more involved or consume anyway more energy, and hence the neurons in these areas are likely more activated than all the other neurons in the background of the brain. So now using these two technologies, which we just saw, we can do what we call connectivity and look at the different groups of neurons that are activated during a specific task and their connections, and hence reproduce what we call neuronal networks. Now, I just need to specify one thing, though. These are models. They are fantastic technologies that produce amazing images, such as the one you see right now, but these are only models, and the actual resolution of these models is order of magnitude below the reality. We'll come back to that a bit later. But using these two approaches, we can construct what we call connectivity map and study large-scale cognitive networks. Now, there are different models with different number of cognitive networks out there in the literature, but I think we can state that there's at least 7 basic networks in the human brains that have been highlighted thus far. And our interest will regards only 3 of these networks. They are the 3 networks involved with the general circulation of information in the brain. So the first one is called the salience network. The other one is the default mode network. And finally, the central executive network. Now these guys are organized in a kind of a hierarchical fashion. So the salience network is like the general, the trigger, the one that will decide whether you are in central executive functioning or in default mode networking. You can't activate these 2 networks at the same time. It's either one or the other, and this is decided by the salience network. So you do see here this organization, and the central executive network is the one that you typically use whenever you perform any kind of cognitive task. So it's the network that actually organizes the circulation of information in your brain. Whereas the default mode network, the one that is shown in red, is a network that is activated when you are losing your time dreaming, thinking of anything but the work you have to do. And this network is actually very important, because it is during these kind of lunatic activities that you think about yourself, think about what you are, what you would like to be, what you are to become. You are ants working on the construction of yourself. And this is a very important network that is, we'll see in the next videos, but that is involved in the genesis of some problems related to digital technologies. So this guy, the default mode network, is very important, and once again, is never co-activated with the central executive network. That is the network of your cognitive functioning. Now, these networks are of a limited scale, as I mentioned, and they are ordered of magnitude lower in terms of precision data as compared to the real situation. This is an example of a paper we published, my group and Dr. Whittingstall and Dr. Descoteaux, maybe 15 years ago, characterizing one of the cognitive network we felt was especially important in our patients with brain tumors, so the visual spatial network. And as you can see, using the graph model, we have 6 nodes and 7 edges. So it's neat, it's interesting, but do we honestly think that it is so simple in the brain? Of course, it is not. And I like to use to give a better understanding of this concept, the analogy of Google. So I tell you, I work at the CHUS in Sherbrooke. So this is where I work on Google. But what is my exact resolution? I work at the CHUS. I work at CHUS. I work at CHUS, and then I work at CHUS. So remember that you have 4 times to the 15 fibers in your brain, and these sets of data can modelize maybe 1 million fibers. So once again, far from the reality. And just to emphasize that point, I'd like to cite this paper published in Science maybe 3 years ago, where scientists of MIT used 1 cubic millimeter of brain in the temporal lobe of a patient that was operated for seizure. And they sliced this sample in 5,000 slices of 30 nanometer to study the number of cells and their connections. They found 57,000 neurons and 130 million synapses in this very small limited volume. So this is extremely important to realize that we are far, far, far in terms of resolution in our understanding of the brain. Okay, these are images from this paper that are absolutely wonderful. And I would like to repeat one thing that we discussed in prior videos on our chain, videos that have to do with neuroplasticity. So do remember that this is not static, it changes during your life. Now, I did my medical studies in the 90s. And in the 90s, when we studied what was the network of speech, well, this was a gross description of that network. So there were basically 3 components to the network. So the Broca's area, the Wernicke's area, and the arcuate fasciculus that links these 2 areas. So nowadays, this is an image from my 3rd book, The Universe in Your Brain. This is the network of speech. So as you can see, it has evolved, it has changed, it is much more complex than we thought it was, and it will continue to do so. My guess is in 100 years from now, this thing will be much more complicated. So I mentioned that only for you to realize that everything we work with has great limitations in the study of the most complex object in the universe, the brain. So I hope you enjoyed that video, and see you next time. So that was it for this week. Thanks for listening to us. We have a lot of pleasure producing these videos on the brain, on neurosciences. It's a topic that we do like. We do enjoy the topic. And the goal is really to simplify matters for you guys. So I hope you enjoy that. You know that it also serves a good purpose in that we are linked to a foundation called Cœur en Tête, which actually support my lab. So my lab does research on brain tumors. We're looking for new treatment strategies. So everything that will be considered a profit on this channel will be sent to the laboratory. So if you like what we do, if you enjoy these videos, do not forget to like the channel, to like the videos, to subscribe, and to click the bell so that you get notifications whenever we get a new video out. Please leave comments as well as questions. If you guys have specific questions on the brain, on neurosciences, and we would be more than happy to respond, to address some of the questions you guys have on the brain. So thanks again for listening. Notice as well that there is a link directly to my internet site that will promote any media activities as well as release of new books. So thanks again for being there.